Hello everyone, my name is Tong Li, a PhD student from Huajin Lab. I'm going to introduce our Journal of Cell Biology paper entitled GxcM-Fbp17 RacC-WASP signaling regulates polarized cortex assembly in migrating cells via Arp2. Adopting a polarized morphology is the basis for a cell to achieve diverse functions, such as nutrient uptake and migration. In accordance with the polarized cell shape, cortical F-actin networks are constructed in an asymmetrical manner as well. However, how their activities are spatial-temporally regulated remains to be fully elucidated, especially for the poor in the realm of migrating cells. Dictyostelium provides a valuable system to study this question. Previously, using the Dictyostelium, we screened for pH domain containing proteins that localize specifically in the real of cells. From that screen, we identified a putative Rho GEF protein GxCM, which is composed of a pH domain, a DH domain, and a C-terminal protein-rich motifs. When expressed as a GFP-fused protein in cells migrate directionally along a chemical gradient, GxCM mainly localized in the rear and the lateral regions. Interestingly, overexpression of GxCM triggers F-actin assembly in the rear of cells, in contrast to the control cells where the bulk of F-actin, marked by Limi-E, is detected in the migrating front. The Limi-E signal is highly accumulated in the rear of cells with GxCM overexpression. Further experiments show that the F-actin induced by GxCM is Arp2 complex-based because it can be marked by the complex subunit Arp4. To understand how GxCM signals to the Arp2 complex and triggers actin polymerization, the different functional domains in GxCM were analyzed. We found that GxCM uses its C-terminus to recruit F-bar protein, Fbp17, which binds to and activates WASP to induce actin polymerization in an Arp2 dependent manner. In addition, GxCM interacts and likely activates the RacC family small GTPs RacC via its DH domain. Constitutively active form of RacC accumulates in the rear of migrating cells and trigger Arp2 based actin polymerization similar to GxCM. Depletion of either Fbp17 or RacC blocks the function of GxCM, causing strong defects in cortical integrity and cell migration. In this study, we delineate a signaling cascade responsible for polarized assembly of cortical actin and maintenance of cortical integrity. This F-actin pool is critical for cell migration and division. Our work uncovers a previously unappreciated function of Arp2 complex-based branch F-actin in building the rear cortex of migrating cells and expands our understanding in polarity establishment. Finally, thank my advisor Huaqing and my collaborators. Thank you for watching.